Welcome to the SB Grid YouTube channel. Software tutorials by developers, lectures by structural biologists, unique content brought to you by SB Grid.
Hello, everybody. Welcome to the SB Grid webinar series continuing. Next month on December 12th, we're going to have Robin Pierce talking about deep fold RNA. And then in January, Graham Winter from Diamond Light Source is going to be joining us to talk about the dials. And today we have a group presentation from Gabrielle Korsko, Hanne Stark, and Bowen Jing. They're here to talk to us about DiffDock, which is an approach for using some deep learning approaches for small molecule docking. And they will explain it much better than I will. So I'm happy to hand it over to them. If you have questions, feel free to use the Q&A function or send messages to one of the hosts in the chat, and we'll moderate until the end. And with that, Gabrielle, Hannes, and Bowen, thank you for joining us again, and take it away.
Excellent. Thank you very much for the nice introduction. And yeah, so we'll be talking about DiffDock here with, and I'm the Hannes guy. This is Gabri and this is Bowen. And yeah, so first we get a little bit into very concretely what is the task that we're considering here, input, output. And then we'll get a little bit into how the method works. And then we'll get into some results and into how to use it in practice. Okay, then let's get started because yeah, the inputs and outputs are very simple. As input, we have the 3D structure of a protein and the 2D chemical graph of a small molecule. So of the small molecule, we do not know the 3D structure yet, and we do not know the 3D structure with respect to the protein. Yeah, and for the protein, we have the whole protein as input, and we're not considering the pocket level docking scenario where we maybe have a bounding box of some pocket that we already know. No, instead, we're doing blind docking where we have the whole protein and want to find out where the small molecule binds. Yeah, and the output of the tool is the 3D coordinates, the 3D coordinates of every single atom of the small molecule. Okay, and there will then also be some further outputs because we can produce multiple candidates, and we will also have a score for all those candidates, but we'll get into that later. But then we wanted to motivate this a little bit. So what do we traditionally do with our usual docking methods, and why now do this with deep learning? Well, the traditional deep learning, traditional docking methods, right? They're based on a scoring function that ranks every single conformer ranks every single 3D position that the ligand can take with respect to the protein. And we have this energy function, the score function, and then we use a search algorithm to search to find the minimum of this energy function. But of course, if we have a very large search space of blind docking and we don't already know the pocket, then this can be quite, can take a long time to find the minimum. And another issue is the sensitivity to slight inaccuracies in the protein structure, such as if we, for example, have computationally generated structures and where maybe a sidechain might be a little bit off. And there has been evidence in some papers that when we're docking to computationally generated, like to AlphaFold structures, for example, then these classical methods, they struggle with this a bit. And with that, we now have our question of what can we do with deep learning for docking? And for that, so far, we've seen regression-based approaches, say where the deep learning method would have some sort of graph neural network where the nodes of the protein are given by the protein residues, the nodes of the small molecule are given by its atoms, and they are also associated with locations. And then we would do some message passing. And for example, this regression approach, it would make its prediction by predicting key points for the protein where where the model thinks that small molecule should bind, or where the model thinks that the pocket is, and it would predict key points or like interaction points for the small molecule, and then we would calculate the translation and the rotation to optimally align those key points and apply the same transformation to the small molecule to end up with the final location prediction. But these types of regression approaches, they did not meaningfully improve the performance that we were able to achieve over compared to traditional docking methods, where here we're in red, we're showing deep learning methods, and in blue, we're showing traditional search-based methods. Yeah, and now we argue that, or we have this little summary here where we argue, right, in our search-based method, we have our energy function, we learn this ground truth energy function, or not ground truth, we learn the scoring function, and then we use a search algorithm, right? We start somewhere at a random location, and then we use our search algorithm to find the modes of this energy function, or the minimum of this energy function. But we might very easily get stuck in local minima. Meanwhile, if we have our deep learning regression approaches, and I should also mention here in green, in this visualization, we would have the ground truth, like what we want to predict, and in yellow we have what we get as output with the method. And so this is for the traditional search-based methods, and then here we have our regression-based deep learning methods, where we have this distribution, right, which during training we sample our data from, and then we make our prediction with our model, and our prediction tries to minimize the mean square error. And then the best it can do, if it makes a single prediction to minimize the mean square error to samples from this distribution, is to put its prediction at the mean. But this is actually not what we're interested in here, right? We're interested in the sample from the modes, the global mode. And this is why we argue another approach, generative modeling, should be the approach taken for the docking problem. And that's what Gabri will talk about now.
Okay, so Hannes has motivated kind of why traditional docking methods really struggle with a very large search space, why kind of the previous deep learning methods based on regression also struggle in this task. And to give another kind of intuition for why this is the case, what's particularly hard about docking, and what's particularly hard is that there is a lot of uncertainty in the task. And this is both aleatoric, which just means that there might be multiple poses, and epistemic, which basically means that the model will be undecided between multiple poses. And if we have regression models, as I'll show you on the next slide, we're going to get like some kind of mean that's not very useful, while with our generative model, we'll try to populate all these modes. And let me give you a couple of concrete examples. So here we have this protein in gray. This is actually a drug target against malaria. And you can see in green this chromin inhibitor that docks at these two sites in the proteins. And if we run one of the regression models, we obtain a prediction that is right in the middle. This is clearly not a useful prediction, while with the generative model that we'll show, we are actually able to sample both modes. Similarly, we'll see we have here another complex where instead we have a single true docking pose. And however, still these regression methods really struggle, either putting a large part of the ligand in steric clash with the protein, or having this completely unphysical conformation. And instead, we will see that we are actually able to sample relatively accurately the pose with a generative model. And Bowen will kind of introduce how we are actually constructing this generative model.
Okay, we don't need to switch. Oh, okay. All right. All right, so I'll briefly talk about the mechanics of the model and how we actually have this generative model for molecular poses. Now, there are many different classes of generative models in deep learning, and what we're going to use is diffusion models. These have recently been quite famous. If you've heard in the news about AI-generated art or photorealistic imagery, this all came from diffusion models, which are very good at modeling very complex probability distributions, which makes them very well suited for molecular structures as well. And it's this aspect that we leverage in developing DiffDock. Now, I want to briefly outline how these models work, just to ground everyone in some common language. You've probably heard people say that diffusion models add noise to the data and then learn to remove noise. So what that kind of looks like is you have your data and you can imagine some kind of diffusion process happening here. So you can imagine red is like concentration of the data at a particular point in space. And this diffusion process is adding noise. And then with your neural network, you learn a vector field that points in the direction of higher concentration or probability density. And this intuitively is the part of the diffusion framework that corresponds to removing noise. And this vector field is going to be evolving over time, right? So it's a pretty complex function that we're going to approximate with the neural network called the score function. And then what you do at inference time is you draw random samples from your initial noisy distribution and follow the neural network as it tells you how to remove noise by following this vector field until eventually you get to the data distribution. Hopefully the visualization is clear, even though the arrows are kind of small. So now this right here is just a toy diffusion on a 2D space. What we're going to want to do in DiffDock is think about how this generalizes to the space of ligand poses, right? So just to emphasize that even though we're talking about diffusion with very similar mathematical formalism to physical or chemical diffusion, this is really diffusion over the space that the data distribution lives in, right? So in the case of ligand poses, we're going to want to think about what that space is and how to diffuse over that. So the space that we're actually going to look at is actually quite inspired by the way that traditional docking methods have thought about the space of ligand poses. In Glide or Vina, you're probably familiar that you provide a conformer of the ligand as input. And then what Glide or Vina will do is it will move this ligand around with rigid body motions and update its torsion angles, but it will not disrupt the bond lengths, bond angles, and ring structures of the ligand, right? So what we're going to take away from this paradigm is that the space of ligand poses is actually this non-Euclidean manifold that is described by the space of poses accessible by twisting these torsion angles and moving around the ligand. So this is going to be the space of ligand poses that takes the place of that 2D toy Gaussian example that we saw on the previous slide when it comes to thinking about diffusion models. So the diffusion that we're going to want to construct, the kind of noise that we're going to want to add is going to be this kind of like chemically consistent noise with the initial RDKit conformer, right? So the noise is just going to be in the ligand structure, rather in the ligand torsion angles and its rigid body motion. And when we talk about doing diffusion over this space, what we're going to do is train a model that removes noise of this kind, right? This is going to be a model that removes torsional noise, positional noise, and orientational noise from a randomly seeded initial ligand pose in order to move it towards the distribution of the true pose. And so I will skip these technical details here, but what that really looks like is shown in the bottom left corner here. So recall in the earlier visualization that we had a neural network that tried to learn a vector field on this 2D space so that it could point towards the direction of lower noise, right, in the direction of higher probability density. That's exactly what's going to be happening in the score model that we have in DiffDock, except that the noise is torsional, orientational, and positional, right? So what that means is that the score model is going to look at the current ligand pose, and when I say current, what I mean here is because diffusion is an iterative generative process, right? So the input, the beginning of a diffusion generative process is going to be some randomly positioned ligand pose that you're going to just initialize the diffusion process with, right? And the assumption is that this random pose has a lot of noise, and the model, the generative model is going to be progressively removing that noise one step at a time. And there are three kinds of noise: positional noise, translational noise, and torsional noise. So the score model is going to predict a vector, this brown vector here. So that's kind of like the direction that of removing translational noise. You can think of that as like a linear velocity or linear momentum. We're going to have a rotation vector, which is removing orientational noise. This is like an angular momentum or angular velocity. And then for each torsion angle, we're also going to predict a quantity that tells us how quickly to twist that particular rotatable bond and in which direction in order to make the pose look less noisy. And this is, I guess you can say it's like an angular velocity around that particular torsion angle. All of this is done with a particular kind of message passing neural network, which I will not get into the details of. But the upshot of all of this is that the generative process looks something like the following. So again, similar to Glide or Vina or Autodock or any of these very well-established docking tools, the input to our method is again going to be a conformer, either from RDKit or maybe from the Cambridge crystallography database if you prefer that. And what our model will then do is it will first sample from the equivalent of that initial Gaussian distribution, which in this case means that we're going to randomly position the ligand relative to the protein. We're going to completely randomize its orientation and its torsion angles. And so the distribution of poses looks something like what you see at the bottom left here. And what this corresponds to is kind of like the noisiest state possible for the pose. And then we're going to just progressively use our model to figure out how to remove noise from the pose, both translationally, orientationally, and conformationally by adjusting the torsion angles. Do this multiple times, in practice about 20 times. And then all of the poses will hopefully move towards a low noise state, which hopefully corresponds to them being in the binding pocket. Now, I do want to emphasize that here we are showing independent samples and independent trajectories, right? So when these poses are moving relative to each other, they are not interacting with each other in any way whatsoever. This illustration is just showing multiple instantiations of that process. Now, of course, finally, you do want to have an ability to select from this distribution of poses a high-ranking pose for downstream analysis, and we also provide a bespoke confidence model. This is trained as a under 2 angstrom RMSD classifier, and it will it will select out from the many samples that you can draw from the generative model which pose you would use for downstream analysis. Here is just kind of a final visualization of that. So what we have here at the beginning is a cloud of randomly initialized ligand poses, and then as you can see, they move towards the binding pocket. One thing that I do want to emphasize here that is quite interesting, and maybe this is a key point of difference between this iterative process and a traditional docking process, is that as you can see, during the course of this denoising, the ligand oftentimes will pass straight through the protein, right? It will look like it passes through very energetically unfavorable regions of state space. Now, this is a good thing because this is what allows us to actually reach the global minimum of this data-driven energy function in the first place, right? Because if we otherwise had to deal with this rugged energy landscape, we would not get there. But the other aspect that I want to highlight is that DiffDock is not trained with any notion of steric clash, right? It is trained only with the objective of getting as geometrically as close to the ground truth pose as possible. So what this means in practice is that the output pose, for example, if you're doing cross docking or if you're docking to an AlphaFold structure or a apo structure where the side chains are wrong, DiffDock will put the pose in what it thinks is the right binding pocket, but without any regard for whether or not it clashes with the side chain. So when you look at a DiffDock pose and evaluate whether or not you like this pose or not, the energy or the scoring function under a Vina scoring function, for example, will probably not be the best metric that you will want to use. You'll probably want to do some relaxation first, because DiffDock, while it will generally get the geometry of the pose right, it will not try to do anything about these energetics. So that's something to keep in mind. Then I will hand it over to, who is it this time? Gabri, to talk about results.
Okay, and if there are any questions on the method side, I guess we can take them also now, but otherwise, also happy to take them at the end. Okay, so let's see some results and some summaries. So first of all, what do we train this on? So we use PDB-Bind, which is standard benchmarks. It contains about 19,000 structures from PDB. They are being curated to be of higher quality and obviously containing ligands. We do a time-based split, so we train on complexes that was resolved before 2019, and we test on newer complexes. The complexes that we test on, we make sure that there are no ligands that were in our training set to look more for some kind of generalization. And we have various different kinds of baselines, both from traditional methods and deep learning ones. Okay, so the first set of results that we are going to see is when we provide to the methods the hollow structures. So this means that we actually feed into the methods the exact structure that the protein will take when it will be bound. And this is typically the way that these methods are evaluated, although one could argue that it's not very realistic. But we can still see here that for this blind docking task, traditional methods and these deep learning methods don't really get a success rate, where here we're measuring success by the proportion of predictions with the RMST, top 1 RMST below 2 Armstrong. And we can see that the success rate is below 25%. Now, this can be increased by combining basically a pocket finding methods like P2Rank or Equibind itself with one of these traditional methods focused on that specific pocket. But then we actually see that DiffDock itself can achieve a significantly higher performance. We can also look at the performance on predicted structures. So this is in the setting that instead of feeding the ground truth structure, so the ground truth bound structure, we're feeding the ESM fold generated structure. So we feed the sequence of the protein in ESM fold, we get the structure, and then we try to dock the ligand to the structure. And as Anna said at the beginning, the traditional methods have also been shown in previous works, but we can also see here that they really struggle in this task. The performance and the success rates drops all the way to below 5%. And the reason is because, often the side chains of these structures are, not accurate, by the sense that, they will change upon binding. And on the other hand, you can see that actually DiffDock is a lot more reliable and loses a much smaller percentage of its success rate. And so it retains some good level of performance also on AlphaFold or ESM-fold predicted structures. Now we actually wanted to kind of give some shout out to some of the works that have used DiffDock since we published. So this, for example, is a very interesting work on where DiffDock was basically used to do reverse screening. This came from the group of Tim Peterson at the Washington University in St. Louis, where they use DiffDock and trying to bind DiffDock on a series of proteins on a particular pathway to try to understand the mechanism of action of a new drug that they had discovered. And this is a very promising, for example, application where blind docking, and in particular blind docking to AlphaFold or in general like APO structure, we think will have a big impact. To kind of summarize a bit more, kind of using kind of the tool itself. So you can find, first of all, kind of more detailed in structure on our GitHub, where you also find the models and some Colab notebooks in case you prefer using those. And so the input is a protein structure. And here you can either give the structure file if you have, like, for example, a crosstalk structure, or you can even feed the input, and in which case the model will fold and the protein with ESM fold. And then you have to provide the ligand. And so here again can be either a structure file or a SMILE string, but in either setting, we don't assume that this structure is actually accurate for the ligand. And then the reverse diffusion runs. We run the confidence model, and then the output will be basically the files with the predicted ligand poses, where you will also find the rank and the confidence of each of those poses in the name of the file. And this is kind of mainly what we had. We also have wanted to give a brief overview of a follow-up work that we are going to be releasing soon called DiffDock Pocket, where we tackled two of the commonly reported problems with the original DiffDock for many people that have been using it. And one was the ability of not being able to control for a specific binding pocket. And the second is, although I've shown you some good apostructure performance, there was not really any way of predicting the rearrangement of the side chains upon binding. And so, potentially, making the relaxation step that Bowen was talking about harder. So what we do here is we do a few changes to have this pocket conditioning, where we restrict the focus to the pocket and we give access to the model to the full atomic coordinates. And then we also add side chains torsional flexibility built directly into the diffusion process. And again, you can see here, again, kind of holo docking performance on PDB bind when conditioned on a specific pocket. So when we are dealing with holo structures or often with cross-docking structure, we are at least as good or better than some of these traditional methods that were designed for this task. But again, we have significantly better performance when docking to apostructures. And we can see here also when it comes to predicting the correct rearrangement of the side chains, we do significantly better than some of the traditional methods. And with that, we'd like to thank you for listening and open the floor for any question.
Thank you. It was a very interesting talk, and I have to admit that DiffDock Pocket answered one of the questions I had queued up before I even got a chance to ask it. So, well preparing. So we have one question that came in from Joseph DeCorte. Beyond relaxation, do you recommend local refinement with one of the DiffDock pose with one of the more traditional algorithms?
Yeah, so I think we've people have used DiffDock with a combination of different tools. In general, it kind of, I think, depends on kind of what you're gonna use kind of down the line. I think in general, whenever you're using some, for example, scoring function or some energy function, it's best to basically either relax or do some pose refinement using that same scoring function. Because different scoring function and also the intrinsic scoring function of DiffDock have kind of slightly different properties, and so in general, depending on the tool, I think that you want to use downstream, you should basically do relaxation with the same kind of level of tool.
Thank you. And I guess, since you touched on the sampling a little bit, is that a case where you've tested multiple different ways of doing the scoring during sampling, or could you just say a little bit more about how you're doing that?
Scoring during sampling? What is meant by that?
So when is that just purely geometric, or is that one of the traditional atomic force fields?
So the sampling process itself is not driven by any physics-based energy or scoring function. Oh, are you talking about this part, this ranking of the poses?
Nope. I think going back further. So you were
Okay.
answering the right area. So
Yeah, the actual sampling itself. So I mean, so you know, as I kind of overviewed here, in diffusion models, the neural network is actually predicting the movement that would bring the pose towards the data distribution. So rather, maybe that was not the best illustration. Maybe this is the slide. Sorry for so many slides. So in this slide here, the neural network is quite literally predicting a set of two vectors and a torsional velocity around each one of these. So it's all coming from the neural network, and it's all data driven. There's no physics-based sampling involved.
So is there a way to incorporate any reliability information about the protein structure into the DiffDock way of thinking about it?
So in general, the question of how to incorporate this prior knowledge into these diffusion sampling processes is an active area of research.
Yeah, there are lots of interesting ways to go here, and yeah, I was just thinking, like, what if you have B factors? What if you have multiple AlphaFold or ESM fold predictions, and you want to use a cluster? And sounds like I should wait and see for future research.
Yeah. I mean, one very first pass idea that one could have is if you want to restrict to a certain binding pocket, then you can like manually adjust this translation vector. So that's the one in brown here. So one thing that you can do if you want to dock to a specific pocket without retraining DiffDock is just, you know, if the brown vector is pointing towards a different pocket, just, you know, correct it. But what one generally finds is that when it is possible to retrain the model to explicitly use the prior information as input, it generally does better. So maybe that's why the focus has been a bit more on how to incorporate the different kinds of knowledge that you would want to incorporate. But it is true that there are a number of inference time tricks that you could consider doing. And the rough paradigm is just, you know, like you can manually adjust any of these updates if you feel like they're wrong.
Well, one other approach for, like, if you wanted to target a pocket before DiffDock Pocket came out, it sounds like you could just edit your input structure and delete the parts that you didn't want to have docking on. And that would be ugly and feel kind of like a hack, but.
Yeah. And, you know, here, you know, it's again, there are different ways that one can think about doing it. I mean, one could also like just sample multiple times and still you get poses that are inside that pocket and, you know, do a manual kind of confidence filtering. But in general, you know, that's not really kind of making the model kind of like, you know, we would expect the performance when, you know, restricting to a particular pocket to be better because the model should have an easier task. And so this was one of the motivation for actually, you know, training a model fully on the pocket and, you know, also trying to predict, you know, pocket rearrangements at the same time.
Yeah, thank you. I guess another question, could you talk a little bit about the general benchmarks for as far as speed for like number of molecules per day for GPU? And obviously there's going to be a lot of range, but.
Yeah, so let me see. I think it might be on one of the supplementary slides. So it's always a bit hard to compare these methods. So first of all, so here it's kind of the general number, which is like on depending on how many samples that you take when you run DiffDock, it takes either 10 or 40 seconds on the GPU. And so, you know, you can, you know, based on that kind of, I guess, do some calculations on kind of how many samples a day you can obtain. There are a range of tricks that one can do to accelerate them. And, you know, the obvious one is you can take fewer samples if you need to kind of, if you have access to less computational resources. And you can see in our paper, or I think we should also have here, that there is a sort of, obviously a curve. So, if here we've kind of presented results with like 40 samples, but you can, you have relatively little loss in performance just taking 10 samples. And that's, for example, 4 times faster. So, there is a range of hyperparameters that one can play with if speed is it's an important consideration. And but yeah, these models also, something I should say is that they do run best on GPU. So running on CPU often takes even longer than the traditional methods when compared on CPU.
And could you say a little bit about the VRAM requirements, or any, because some things are very VRAM heavy, some things are not, and that can, nobody has enough GPU time.
Yeah, so it really depends, to be honest, on the size of the protein and the size of the protein and the ligand. But something that can be controlled and is one of the hyperparameters that can be fed into DiffDock is basically the what we call the batch size, which is basically how many complexes are sampled, how many kind of samples we take in parallel. So basically, if you're taking 40 samples, by default, we're taking them in batches of 10 samples. But if you have a smaller memory, you can scale this to, for example, to 4 or 8, and to fit your GPU memory.
Thank you. We've got a couple more questions coming in. So could you say something about it, if the scoring for DiffDock Pocket is that, or DiffDock in general, how would you compare that to traditional scoring? And I believe this is something that you addressed earlier, but I might be wrong about that.
Well, I think maybe to answer that question without further knowledge of what specifically is meant by the comparison, the scoring by this confidence model here is based on a trained classifier where the model is trained to predict whether the pose is under 2 angstrom RMSD or above 2 angstrom RMSD. So what that will mean is that compared to like a traditional scoring function that's based on pairwise interaction terms, this scoring function will give a very good score to a pose that is, say, 1.5 angstroms away, but has overlapping atoms with one of the side chains, right? But it will not give a good score to a ligand that is in the wrong pocket, but happens to have a very good energy inside of that pocket. So I guess maybe to summarize and to hit home the message, this scoring function is trained with the sole purpose of selecting poses that are within 2 angstroms RMSD geometrically of the ground truth ligand pose. So it is, if ideally trained, it is a convex energy surface, right? There will be no spurious local minima. Of course, that may not be the case in practice. But this is to sharply contrast with traditional scoring functions, which can be very, very sensitive, for example, to specific interatomic distances, avoiding steric clashes, and have a very rugged energy surface. Hopefully that answers the question.
I think it does, and it illustrates the, is A better than B? Well, better for what? But
right, exactly, yeah.
And Jason has his hands up, so I assume he has another question.
Yeah, I did. If you're taking this approach where we're not really paying attention to clashes, we don't really have to worry about physics. We got a big set of conformers. We just dive in and let the. What are the ramifications for the size of the ligands that you can sample? Because can you go to bigger conformational spaces? Can you use say more rotable bonds? Does it have? Have you looked at things typically with the approaches people use, they use fragments or maybe even like, 20, 25 atoms. But can you go even bigger with this? And do you pay a penalty performance wise?
Yeah, so definitely that's a great point, which is the fact that you're using this kind of smoothed out energy surfaces is particularly gives particular improvement when your dimensional space that you're searching over it's higher dimensions, and so we do expect it to do better in terms of when you add more and more kind of degrees of freedom. Now I don't know if we have some of these results in the presentation, but also the other problem, however, perpendicular problem, is the fact that, potentially larger and larger ligands are kind of out of, the domain where, the model was trained. So that's a potential caveat. But I think one setting where it becomes very clear kind of disadvantage is when you look at predicting flexibility of sidechains. So, also many of the traditional methods allow you to basically enable some flexibility in sidechains by basically allowing to also model the torsion angles of the sidechain, similarly to the way that Diffdog Pocket does. But when you actually see how these models do, they actually work pretty terribly. And I think from my intuition, the reason why kind of they really struggle is that the degrees of freedom really increase, and so this basically search algorithm that they use kind of really struggle. And so this is, I think, kind of one setting where we can really see the advantage of having this kind of like diffusion-like approach versus energy-based approach and search-based approach of traditional methods.
Yeah, that's an interesting. I hadn't thought about that. That the diffusion-based approach, well, it kind of skirts that limitation in the sense that it doesn't have to worry about like steric interactions, is limited that it's trained on it. So like it's expanding beyond that is probably challenging. Thanks.
I think that may have run through all of our questions. So if there are no more from the audience, then thank you for a great talk, and I enjoyed it, and I hope the audience did too.
Thank you very
much.
Thank you very much. It's great.
And I guess maybe one final question on a lighter note. You'd mentioned the image generation AI tools. Do you think that the intuitions that people would develop playing around with those tools would transfer to something like Diffdoc in terms of, oh, I need more sampler steps, oh, I need a different batch size, or is that just,
Well, I mean, it is the same kind of model. So at the end of the day, there's a ton of shared language. And pretty much every concept that has been applied in image diffusion has some kind of analog in molecular diffusion. And if they haven't been explored already, then they're active areas of research.
Cool. Well, thank you again.
Thank you very much.